let's make a start then. Thank you everyone for joining us tonight. I appreciate that it is a very hot evening, so really appreciate you taking the time to attend this session with us tonight, and we really hope that you do enjoy it and find it informative. So the event tonight is the Research Lounge, the future of gastrointestinal and liver research. The Research Lounge is basically put into place to encourage conversations between our research and our patients, public and wider Nottingham population. I am Kate Frost. Almost forgot to introduce myself. So my name is Kate Frost and I'm the head of patient and public involvement and engagement for Nottingham University Hospitals, and I cover all the research that happens across Nottingham. So what I'm going to do now is just run through the agenda of the session very quickly. There we go. I hope that has changed slides for everyone. Yep, I can see some nods. Excellent. I'm going to, in a moment, pass on to the theme lead of gastrointestinal and liver research at Nottingham Biomedical Research Centre, Guru Afal, who's going to introduce himself and then pass on to the speakers for today's session. Following that, we've got three very short but snappy and hopefully very interesting presentations. The first is by Dr. Karen Robinson, and it's going to look into new technologies exploring the infection causing stomach ulcers and gastric cancer. So that is around diagnosis and treatment. Following this, we are going to be joined by Jane and Mel, who will be doing a presentation on what do you do with my blood, pee and poo. So this is going to be talking about taking science beyond the lab, translational research in liver disease. And then this is going to follow by Nav, who is going to be discussing imaging methodology in use, such as magnetic resonance imaging. Following this, we are going to, and thank you everyone who signed up via Eventbrite, you should have selected your breakout rooms of choice. So what I'm going to do is do my best to manually assign you to. If you selected one, then it'll be that room of choice. If you selected more than one, it'll be one of those rooms. So you'll be automatically pulled off into these breakout rooms for half an hour of a discussion around that theme of choice. And here you'll be able to see our wonderful facilitators who will be helping us with those breakout room sessions. And then briefly, you'll be pulled back into the main room where we'll say thank you for your attendance and then next steps. So that is it from me. If that's okay, Guru, I will pass on to yourself for a brief introduction and stop sharing the slides.
Hello, I'm Guru Aital. I'm Professor of Hepatology in the University of Nottingham and School of Medicine. I've been member of this research group since the beginning of Biomedical Research Unit, which started the process about 2007-8, and we set up first Biomedical Research Unit in 2009. This is seen as the most prestigious and valuable program which is funded by National Institute of Health Research, which is also very new, was conceived in 2007 and is now the biggest body which provides research funding to researchers of a lot of kinds, mainly human-based research for the whole of coverage of NIHR. And most of my colleagues today who are presenting have been in the area for a long time, but some of them, I would say majority of them, have been there or thereabout with us since 2009. This is the fourth cycle we are going to get funded. Just to highlight the process is that we are asked to present a program of work in what was decided by NIHR on what are the important areas. They decided the importance or in need by looking at what the burden of the particular health problem is in the UK and what was the amount of research funding allocated in the UK. And where there was a gap, they are identified as an important area. So as identified them, we matched our strengths and applied on the main area of benign conditions in gastrointestinal and liver disorder. And that still remains our main area of strength. So what we have done so far is go through 3 programs of this kind. Each 5 years, we need to rejuvenate ourselves, apply for a new set of ideas within the broader remit of benign gastrointestinal conditions. And international panel, purely international panel, will look through our proposals and advise NIHR about the funding, which means that we are judged in an international level, not in the national level. What we are talking about today are the mixture of 2 things. We always had an open meeting. We always wanted to keep in touch with patients and public, because NIHR's program, particularly BRU initially and BRC now, is focused on what's called an early translational research. Before NIHR born, it was seen that there was a gap between how good the discoveries happen in UK, how well it's funded in UK, and how the NHS care changed in UK, which meant that more we find, we weren't implementing into clinical practice. So the early translation is what all of the group are involved in as of today and through the BRCs. Early translation comes from discoveries in science and first time or an early phase of how it is applied to humans, either people or patients. And we used to have an open day to show how we think of an idea, how we implement, and what each of our skills are. And from COVID-19, that has been not possible for 2 years. We also had a yearly program called a showcase day. This was for us with the whole group of people who all contribute to research and do the research and lead it, to sit together with a few external reviewers to catch up with what we had done with each other as well as a group. That also hasn't happened for 2 years. So today is the hybrid of it. We usually take entire day, 10 minutes each, to go through the crystal of the most exciting stuff we do. We can't do that today, so you will get a bird's eye view, a glimpse, a flash today. So I will introduce you the next speaker, which is Karen Robinson. Dr. Karen Robinson is an associate professor, has always been at the forefront of research in Helicobacter pylori, a common organism we all have with or without health condition. So Karen has researched on both the good side of having this bug inside as well as the bad of it. So over to Karen.
Thank you. So thanks very much for taking a bit of time out on this really hot day to listen to what I've got to say. Hopefully you'll find some of it interesting. So for many years now, I've been studying this bacterium called Helicobacter pylori. And you can see a picture of these little beasties in the corner here. So they've got these whip-like flagella at the end that helps them to move around. And unfortunately, they cause stomach ulcers and gastric cancer. So I'm going to start by telling you a bit about sort of where we're at the moment, what the problems are, and some ideas that I have that I want to put in with the new BRC application. And I really want to know what patients think about these ideas, whether they would be prepared to take them, or if there are some problems that they can see. But maybe we need to change our thinking a little bit. So I'd be really interested to hear your thoughts. So where am I? I've lost here we go. There we go. So Helicobacter pylori is one of the most common infections in the world. Nearly half the population of the world have it, and they don't know it for the most part. So they get it when they're a young child and they keep it for their lifetime. But in about 10 to 15% of cases, they develop disease. So this bacterium stays in the stomach and it can cause ulcers in the stomach and the duodenum, and it's a leading cause of gastric cancer. And gastric cancer is a really nasty diagnosis to have. In the UK, about 40% of people who have that diagnosis die within a year, and the 5-year survival rate is really low. And that's because of it takes a long time before the symptoms develop. People never knew they had it, and then all of a sudden they've got these nasty cancers. So the top here, I've got some pictures from endoscopy. So we've got a normal stomach on the left, and then a picture of an ulcer in the middle, and you can see the mass at the top there. That's a tumour in the stomach. So when people first get infected with this bacterium, it swims really quickly out of the acid in the stomach into a thick mucus layer in the stomach lining. And it could stay there for life, usually. It doesn't penetrate through the epithelial layer in the stomach. So these cells in the stomach lining form a tight seal to stop acid and things in the contents of the stomach from damaging the tissue. So it's really specialized, but it does provoke an inflammatory response. And you can see in this diagram, we've got inflammatory cells coming through. And it's really this inflammation over a long time, because people keep it for years, that causes disease. So people who've got chronic symptoms, chronic dyspepsia, so they've got upset stomach that won't go away, they might be referred to the hospital by their GP for an upper GI tract endoscopy. And during that session, biopsies are collected from their stomach lining, so small bits of tissue, and a rapid test is done. And if they're positive, they're given a triple therapy of 2 antibiotics and an acid suppressant drug. And so the most common combination is amoxicillin and clarithromycin with omeprazole to reduce the acid in the stomach. And if people are allergic to penicillins, then they can't have amoxicillin, so they get metronidazole instead. Now, antibiotic resistance is becoming more common in these bacteria, but no testing is done. So people are just given a first-line therapy, either one or the other combination. And if we've got resistance happening, then that might not work. And sometimes people have to have several rounds of therapy. And in fact, it's policy in the UK that antibiotic sensitivity tests are only done when therapy has failed twice. And there's no routine surveillance, so nobody really knows what's going on. So we had a look at what the resistance profiles were like in Helicobacter isolates gathered from patients in Nottingham between 2003 and 2020. And you can see here, so these are the main drugs that people are given. 96% were resistant to metronidazole, 28% were resistant to clarithromycin. So that's really bad. But very low numbers were resistant to amoxicillin, levofloxacin, and tetracycline. So that means if you're lucky enough not to have penicillin allergies, then you're probably going to be in a better position. So this antibiotic resistance picture is getting worse. And in 2017, the WHO listed clarithromycin-resistant Helicobacter as a high priority pathogen. We need new drugs. But it's not just the drugs themselves and whether there's resistance. There are other issues. So you might think you take antibiotics, these bugs live in the stomach, the antibiotics are in the stomach, so they work locally, but they don't. Instead, they've got to be absorbed into the bloodstream and then go back across this very tight layer of cells, the seal that I was talking about before, and into the mucus layer where the bacteria live. So I've just put these blue blobs on there to represent where the bacteria are. So you can imagine it's really inefficient to treat this bacterium. It's hard to do it. and not many antibiotics can be secreted. So it really limits what we're able to do to treat patients. So what can we do? So the projects that I'm going to tell you about now very briefly are, first of all, we want to try to get drugs to work in the stomach so that they can kill Helicobacter more efficiently. They don't have to work through the bloodstream and then go back across that layer of cells. And we want to make it easier to isolate the bacteria so that antibiotic sensitivity testing can be done and patients given the best combination of drugs. So at the moment, the only way to isolate Helicobacter is through giving patients an upper GI tract endoscopy. So you can imagine people don't necessarily want multiple rounds of having endoscopies and tissue samples taken. So in project 1, we're developing a system to deliver antibiotics that will act in the stomach, and we're using these liposomes. So liposomes, if you imagine a very scaled up version of that, would be like a balloon full of water. So there's a lipid membrane on the outside and an aqueous solution in the middle. So we can put water-soluble drugs like amoxicillin in the middle and things that aren't water-soluble in the outside layer. So these particles are small enough to go through the mucus. We can put antibiotics in the middle. We can put some other fatty molecules in the outside layer that have got antibacterial properties as well. And because it's like a membrane, when they meet Helicobacter, they fuse with the membrane of the bacteria. So they're going to deliver the antibiotics straight to the bacterium itself. And if we can get more antibiotics delivered in the stomach, then we won't have so much going into the intestine. And that's what causes patients to feel bad after they've taken antibiotics. We don't want to affect the gut flora. Okay, so we're also going to make these particles with the antibiotics inside, and we're going to coat them with molecules that mean that they're going to stick to the stomach lining. So they're going to be right where the bacteria are. And we've started to screen what we could use in the outside layer to give a bit more bactericidal activity, so we can get better killing. And we've screened some natural fatty acids, and we found that linolenic acid, which is present in nuts and seeds, can kill Helicobacter. And if we put linolenic acid in the outer layer of those liposomes, we could kill Helicobacter. So these are untreated bacteria. So we've got about 20 million bacteria in a mil of culture. If we put liposomes in there without the linolenic acid, it did nothing. In fact, they seem to like it. We've got more bacteria. And then we've put in a treatment with the liposomes containing linolenic acid. And you can see that if we put 100 micrograms per mil of linolenic acid in those liposomes, we've got killing. And the liposomes on their own don't do anything. So that was really good. So we found a fatty acid that works. We also wanted to look at whether that fatty acid would help to make the liposomes merge with the bacteria. So we've done an experiment where we made liposomes with and without linolenic acid. We put a red dye, a red fluorescent dye in the membrane, and we added those to green fluorescent bacteria. So in this method, you get bacteria going through a stream in single file, and it takes pictures of each bacterium or each cell, and it takes a picture with the green fluorescence, a picture with the red fluorescence, and we've got a merging here. So on this side, we've got the liposomes with linolenic acid, so that's the red. So we've got the bacteria, we've got the liposomes, and we can see it's merged. So we've got both in the same place. On this side, we've got liposomes without linolenic acid. So we've only got the green bacteria. We haven't got any red in this. So you can see that not only does it kill them, but it helps to make them merge together. And in this final slide here, so we've exposed the bacteria to free linolenic acid, or in liposomes, free amoxicillin, and both of them together. And you can see that we've really now got nice, efficient killing with them both together. So we're now up to the stage of working on the molecules to put on the surface to target the liposomes to the stomach. We're looking at how well it goes through the stomach, through the mucus layer in the stomach, and we're making formulations with multiple antibiotics now, not just amoxicillin. Very, very quickly, we're also trying to find a way to recover live Helicobacter without needing an endoscopy, so that we can profile whether they're sensitive to the antibiotics. And our vision is to go fishing for Helicobacter using a capsule that contains a small sponge, and it's attached to a string. So you swallow the capsule, and then that allows the sponge to expand. The sponge is coated with polymers that are going to bind Helicobacter really efficiently. And then you pull the sponge back up through the mouth with the string, and then you test whether those bacteria are sensitive to antibiotics. So you get the right combination of antibiotics in the first place. This is already in place as a way of diagnosing Barrett's esophagus. And there's some pilot data to show that you can recover Helicobacter using this, but the efficiency is low. So what we want to do is improve the binding by adding polymers to the sponge, and then using this as a way to isolate Helicobacter without needing an endoscopy. So we've started screening. So we've got all these polymers on a slide, on a microarray, and so far we've selected 8 polymers to study. Hits from the first screen we're going to put together to use a second microarray with combinations of polymers. And once we find the right combination, we're going to validate those to make sure we've got really good binding, and then use it to coat sponges. So, as I said at the beginning, something that I'm interested to know from the audience is whether the concept of the antibiotics that bind to the stomach lining, is that appealing, or would that turn people off, do you think? And the sponge on a string idea to fish for Helicobacter. So it's good because you don't have to have an endoscopy, but swallowing a sponge and then pulling it back up doesn't sound very nice. And so these are the people in my group and the people within the department that we work with. And these are our collaborators, and we've got funding from several different bodies for that. And thank you very much for listening.
So we'll pass on to Jane and Mel, who are going to be giving us a bit of an inside look into what we do with patient sample collection. So Jane, if that's okay, I'll pass over to you to introduce yourselves. Thank you.
Okay, thanks very much then, and thanks for passing on to us then. Hello, then we're going to really talk about a bit about what we do in the lab with samples that we collect from volunteers, and we're going to explain some of the translational research in liver disease that we've been doing in Nottingham. So as Guru said, translational research is really important for healthcare because it turns the work in the lab into benefits for patients through either disease prevention, diagnosis, or treatments. So it's often referred to as this bench to bedside innovation. And really it's by understanding the disease mechanism that we can develop new tests for diagnostics. And we can identify different patient subgroups so that each person gets the therapy that's most effective for them. Liver disease is a huge health problem globally, and it's responsible for more than 20,000 deaths per year in the UK. And very worryingly, it's really increasing year on year. Despite this, there's very little public awareness of the disease, as often it's unnoticed because the majority of people with the disease don't have any symptoms. So earlier disease detection and better management and care have been identified as key priorities to help us tackle this growing problem. The development of chronic or long-term liver disease is generally linked to obesity, diabetes, and alcohol excess, which result in liver scarring called cirrhosis. This happens because the body tries to continually repair the damage that's being caused by repeated alcohol toxicity or by the steady buildup of fat in the liver. But generally, there are no symptoms until either liver failure occurs or cancer develops, necessitating urgent care. Acute liver diseases can also result in sudden liver failure with severe outcomes for patients. We want to find new ways to predict and prevent drug-induced liver injury, which occurs unexpectedly, sporadically in people taking a standard medication or supplement. And although this is rare, this causes a substantial number of liver failure cases. We also want to develop tests to identify people with alcoholic hepatitis who can suddenly develop liver failure, to ensure that they get the available treatments quickly to improve the recovery rate. Our research is focused on identifying the changes associated with disease development through observational studies where we collect patient samples so we can better understand the disease and develop new tests and treatments to tackle it. I'm now going to hand over to Melanie, who's going to explain how the biosamples are processed.
Thanks, Jane. Hi, everyone. So I'm Melanie, as Jane has said, and I'm one of the lab technicians in the BRC. And if you came and volunteered for one of our studies, you may be asked to donate some blood, urine, or stool. And myself or one of the technicians, other technicians or researchers are the ones who receive and process these samples. And you may be wondering, why do we choose to work with these bodily fluids that most people would just flush down the toilet? But actually, there's a lot that we can learn and do with these samples. And so I'm just going to give you a brief overview of what we actually do in the lab. And I'm going to start with urine. So urine contains waste chemicals which has been released by your body as byproducts of biological processes like digestion. And in the lab, we filter the urine to remove any possible sediments and send the urine off for specialist analysis to measure substances that can indicate gut function or disease. Now we actually do a little bit more with blood. So usually when we get some blood, the first thing we do is separate the plasma from the blood cells. And we do this with a machine called a centrifuge, which is pictured to the left here. And this machine spins the blood very quickly and separates the blood components based on weight. And the picture on the right shows a blood bottle before and after it's been centrifuged. And the plasma is the straw-coloured liquid here. So the question is really, why do we care about plasma? Well, plasma contains chemicals and proteins which have been released into the bloodstream by the body's organs. And once we've collected the plasma, we can use certain tests to measure the level of these substances, which means we can test for chemicals and proteins which are linked to specific diseases. For example, the protein CK18, which is linked to liver disease, but Jane will talk more about that later. We can separate the blood further by separating the white blood cells from the red blood cells. And for this, we need to pass the blood through a separation fluid. And the picture to the left shows the blood after it's been separated through this fluid. And the white blood cells are just below the straw-coloured plasma. But you probably can't see that very well. So the middle image shows the white blood cells after it's been separated from the other blood components and grouped together at the bottom of the tube. And the image to the right actually shows what white blood cells look like underneath the microscope. Now, white blood cells are very important for the body's immune system, and there are different types of white blood cells. And we can actually use new technologies to identify each type of white blood cell and the amount the body produces in response to medication or disease. And this can actually help us understand why in some people the immune system attacks the liver and causes liver failure. We can also extract DNA from blood and from stool. And when we extract DNA from blood, we get human DNA. And when we extract DNA from stool, we get microbiota DNA, which is the DNA from all the bacteria living in the gut. And when we sequence this microbiota DNA, we can use it to identify which bacteria is actually present. And I'm just going to give you a very quick overview of DNA extraction. So the first step is to break open the cell where the DNA is contained. And once we free the DNA, We then have to wash it to remove any other cell components. And then we use alcohol to bring the DNA out of solution. And you can actually try this at home if you're feeling a little bit sciencey. And I've included a YouTube link that explains how to do it, or you can just search strawberry DNA extraction on YouTube. So once we've collected all these samples, what happens next? And how do we use science, which can unravel the disease mechanism, to actually improve healthcare? And how do we develop new tests for diagnosing and monitoring people and novel treatment strategies? And with that in mind, I'm going to pass you back to Jane. Thank you.
Okay. So thanks. So as I explained earlier, it's really important for us to develop new blood tests to detect liver injury early, because current methods are unreliable for identifying disease before symptoms appear. The gold standard test of liver biopsy is painful and has risk of severe complications. And actually, it just samples a small part of the liver, so it doesn't tell us about the health of the whole organ. So we're using our scientific approaches to develop new non-invasive tests. For example, pathologists have noticed that in diseased livers, dying cells appear to be bursting open. And since we know that liver cells contain lots of cytokeratin CK18 fibers, shown here in green, which usually hold them in shape, we could hypothesize that the fragments of this CK18 fiber could be detected in the blood of patients if their liver is damaged. So by doing an observational study, you see the graph here shows that the more severe the liver disease, the more CK18 is present and released into the blood. This is seen in patients with this long-term fatty liver disease. If we then look at patients with acute alcoholic hepatitis that often develop severe effects, such as liver failure, we can see that now the levels are sky high by comparison to fatty liver disease patients. We can see that this matches what's seen in the liver tissue, where this smudgy brown stain shows that lots of CK18 is being released from dying cells in alcoholic hepatitis patients. We found this is then a very useful test to potentially indicate disease severity and even predict outcome and responsiveness to treatment. So this could have an important impact by improving clinical decision making. To take another example, research analysing DNA from patients has found that people with some common genetic variations are predisposed to idiosyncratic drug-induced liver injury in response to taking a particular medication. By investigating the gene function in cells and by using computer modelling, we can work out how a tiny change where just one of the DNA letters is different can affect the body's response to drugs. So here we can see how a difference might affect the shape of the encoded protein molecule in immune cells. So just this little pocket here would be affected. This molecule here, shown as a computer representation, is now shown on this diagram as an arm sticking out of the immune cell on the surface of the cell. And because of the variation, this arm can now hold fragments of a drug shown in green that's been processed by the liver cells. By holding on this drug fragment, this can then potentially trigger an overwhelming immune response cascade, which is causing liver failure. This research is useful for clinicians because it means that they can then use genetic tests to improve diagnosis of liver injury being caused by medication, which can mean that the drug cause can be identified and stopped while patients continue taking other medications they need. Finally, I want to mention how information about your gut micropopulation can be useful for understanding your liver health. The bacterial community in your gut has a big influence on many aspects of your health. These bacteria use various substances that you consume in your diet and produce lots of chemicals that can affect your body. For example, they can produce short-chain fatty acids, bile acids, and even alcohol, and can break down fibre to generate substances that your body can use to get energy. So we want to find out if bacteria can promote disease development. and whether dietary changes can improve the gut microbe community and reduce disease development. We've found that people with fatty liver disease have fewer different types of bacteria in their gut, and that certain species seem to be associated with disease. We see a 10-fold increase in the Colon-cellar bacteria and a decrease in the Ruminococcacea, which we think may affect body fat levels and inflammation. Next, we're running clinical trials to see if the dietary modification can change the gut community and improve liver health as a possible treatment for patients. In summary then, we need your blood, pee and poo for research to understand the biology of disease development. Since samples are so valuable for research, in order to make the best use of samples donated, we have a biobank set up that allows leftover samples to also be used by other researchers. Your samples are absolutely crucial to drive the development of innovative diagnostic and therapeutic approaches, so that there is better healthcare for everyone. Thank you. We're happy to have any questions.
So thank you very much, Jane, for that fascinating presentation. I'm sure Jane will be hanging around in the chat as well if anyone has any questions for her. So we'll move on to our final presenter this evening. So I'd like to introduce Nav, who is going to be talking about imaging methodology in use. So Nav, is it okay if I pass over to yourself to do a formal introduction?
Hi everyone. So I'm Nav. I'm a registrar and a clinical lecturer in hepatology, so in liver disease. So what I'm going to do today is kind of give a brief overview of the advanced imaging that we do, both involving the gut and the liver, and I'm going to concentrate on MRI, so magnetic resonance imaging. As you can imagine, it's going to be difficult going through the details or the complete overview of the stuff we do. So this is going to be a very brief, kind of overview of the interesting stuff that we have done in the past. So why MRI? So over the years, Nottingham and MRI have become synonymous largely to the gentleman in the picture here. So this is Sir Peter Mansfield. He was a physicist, was based in the University of Nottingham, and essentially it's his work that has led to the development of MRI and the advanced technologies of MRI. He was awarded the Nobel Prize in 2003 for his work. And the research MRI center in the University of Nottingham, which we use for scanning our volunteers and patients, is named after him. So it's the Sir Peter Mansfield Imaging Center. It's across in the university. To recognize his efforts, he had a tram named after him, and the Castle Rock Brewery also developed a beer named after him. So why is MRI particularly attractive for our patients? So MRI is safe. It doesn't use any ionizing radiation, unlike X-ray or CT scan. So you don't have the cancer risk associated with ionizing radiation. So you can repeatedly scan patients if you want without that risk. So it's safe in scanning patient over a period of time to observe any changes, or to scan patients before and after treatment. So you can scan patients as many times as you want. How does MRI work? MRI uses strong magnets. The magnets causes changes in the tiny particles in your body. And then you kind of measure that changes, that those changes can be kind of go through processing and then they come out as pictures. But more than just pictures, you can get informative numbers from them. So what we call quantitative MRI. So those changes can be actually measured, and you can get measurable numbers that you can then easily measure in terms of what you want to do. And I'll show you some examples as I go forward. So what I'm going to do now is show you a couple of examples in the gut and a couple of examples in the liver of the stuff that we have done over the years. So this is pictures of volunteers who've been in the MRI scanner after drinking a test drink containing different sugars. So these are essentially fancy names for different sugars. And what essentially the picture shows is when you consume these different sugars, they result in different amount of water being retained in your different parts of your gut. Why is this relevant? When patients or volunteers consume food with this different constituent of sugars, they experience different symptoms. So patients can experience bloating or even abdominal pain in patients with irritable bowel syndrome. And what we were able to do is explain their symptoms based on what we found on their MRI scan. So essentially what I'm showing here is with non-invasive MRI scan, we are able to kind of explain what's happening inside their bowel with regards to how much water is retained in each or separate parts of your bowel when you consume different sugars. We are not just able to see static images, so we can see movement in real time. So as you can see, your bowel, your different part of your gut is constantly moving, and measuring the movement is important. So how slow or how fast the bowel moves can explain the symptoms that patients are experiencing. So in collaboration with researchers down in London, our team here have developed new ways of measuring this movement. So as you can see, they have put grid on the pictures, and depending on the deformity, so depending on how curvy or how straight the line is, they can then measure the movement in a realistic way. More interestingly, we can measure how big the bowel is. So the picture on the top right-hand corner is your large bowel, and the different colors are the different parts of your large bowel. So the picture on the left is a healthy volunteer, and the picture on the right is a patient with irritable bowel syndrome, and you can see that their bowel is generally bigger. And I've put the volume there. And then at the bottom, using this new technology with MRI scan, we were able to study what happens in your bowel when volunteers consume kiwi fruit. So kiwi fruit produces changes in their bowel, and the technology from the MRI scan was used to explain the changes that happen in your bowel, mainly with the volume of your large bowel after ingestion of kiwi fruit. So that's the gut briefly in a nutshell. Moving on to the liver, as Jane mentioned, incidence of chronic liver disease is in the rise in the UK. Two main reasons. One is alcohol. Headlines like this don't help. And also fatty liver disease. About 3/4 of people in the UK aged between 45 and 74 are overweight. That results in fatty liver disease. Over a period of time, you accumulate scar within the liver. The scar leads to what we call severe scarring, what we call cirrhosis. And when you get cirrhosis, then you can develop complications from cirrhosis or develop liver cancer. So Jane mentioned, traditionally, we measured the scarring within the liver by getting a liver tissue, so passing a needle into the liver and having a look at the sample under the microscope. Obviously, this is invasive, painful, and it's got complication because it's an invasive procedure. Using an MRI scan, we were able to measure the scarring within the liver. And more importantly, we were able to measure this across the whole liver. So in this graph here, as you get more scarring, the number that we get from the MRI scan went up. So essentially what we were able to show is with an MRI scan, we were reliably able to measure the degree of scarring within the liver. This just shows it in the picture form. The F is just, F is staging that we use to stage scarring within the liver. So more scarring, there's more yellowness in the liver. So this is just color-coded just for the aid of the presentation. We were also able to measure the pressure within the blood supply to the liver. Higher pressure within the system leads to more chances of patients developing complications. Traditionally, we were only able to measure this in an invasive way by passing a catheter down the neck vein into the liver. It's not done very routinely because it's not available in all hospitals. But we developed a new way of measuring this pressure using MRI scan. This is again non-invasive, so it doesn't involve any injection. And we were able to measure this pressure using the MRI scan, and the pressure that we measured from MRI scan here, and this is the gold standard measurement, and there was a good correlation. So there was a good agreement between the two measurements when we did this study in here. We were also able to measure the changes that occurs across different organs. So patients who develop cirrhosis, so severe scarring in the liver, don't only develop changes within the liver, they also develop changes in the heart, in the spleen, in the gut, in the kidneys, and all of these is important in understanding how a patient is going to behave in terms of whether they are likely to develop complications or even understanding how long they are going to survive. But with an MRI scan, with a single scan session, we are able to measure the changes in these different organs simultaneously. So they go in the scanner once, and we are able to measure the changes in the different organs at the same time. And that's really useful in us understanding what is going to happen to this patient over a period of time. So in summary, there's a very brief whistle-stop tour of the interesting stuff that we are able to measure and able to do with non-invasive MRI scan. MRI allows us to measure the structure, so how a particular organ, the gut or the liver looks, how they function. So we are able to look at how the gut moves in real time. We are able to measure blood flow in real time. Because MRI is safe and we can do repeated scans without exposing patients to any ionizing radiation, we are able to measure changes. So we are able to bring patients back after a year, a couple of years, and see what has changed, what has not changed. And again, along the same lines, we are also able to use MRI to study the effect of treatment. So, by doing a baseline scan and then giving them new treatments or existing treatment and repeating the scan, we are able to study the effect of the treatment. And if we find a certain treatment is not effective, then this allows us to give alternative treatment for these patients. Where we want to take this, so we want to be able to translate this interesting novel studies that we have done here, novel technologies, new technologies, into routine clinical practice, because ultimately our aim should be the science, the interesting science that we do, the new development that we develop here should be used to improve a patient feels or improve patients' life. And that should be the ultimate aim of any clinical research. Thank you. Happy to take any questions.
So thank you so much, Nav, it's greatly appreciated. Again, I haven't had any questions in the chat. So if anyone has any they'd like to pose, please do pop them in there now. Alternatively, you can raise your hand, you can use the raise hand icon, or just wave at me if you have your camera turned on, that's fine. Oh, yes. Ramon, yes, would you like to come in?
Yeah, thank you, Chair. The question I was going to ask, which I myself went through the MRI and I freaked out, so I had to be pulled out. So do they give any sedation to calm the patient before they go inside the MRI scan?
So in the clinical setting, so when MRI is done as part of clinical reasons, so let's say if you're a patient in the hospital and you really need an MRI scan for diagnosis or treatment, sedation can be given. In research, we don't routinely give sedation. Obviously, volunteers and patients volunteer to go into the MRI scan, and if they can't tolerate it, then they withdraw the consent and they're removed from the scanner. Giving sedation is associated with a small but maybe significant risk, and we wouldn't want to expose research participants to the risk. At a side point, sedations can also affect the part of the body that we are studying. So sedation can affect blood flow to certain organs. Sedation can affect the way the gut works, and we wouldn't, if we find the change, we wouldn't know whether that is due to the disease or whether that's due to the sedation. So we wouldn't want to add that as another reason. So we generally don't give sedation for research scans, but it is given for patients who have clinical MRI scans in the hospital.
Thank you.
One thing to add, we do now have a wide bore MRI scan in the university. So the issue with MRI, it's claustrophobic. So as you know, it's a tunnel that you go into, and it can be quite claustrophobic. But one of the new scanners that we have is wide bore scan. So the bore is bigger, and that has helped some of the participants in my study certainly to undergo. So I think that will be another way forward.
Brilliant. Thank you so much. We have actually had another question from Andy. Do MRI scans work alongside or benefit PET scans or vice versa? I don't know if you could just quickly explain the difference between the two as well.
Yeah. So, yeah, they're slightly different, Andy. PET scans are looking at kind of metabolically active parts of the body. But we don't do PET scan. I mean, not that we don't do PET scans. The study that I've presented, they're mainly MRI scan and not PET scans, basically. But they do, and they show different things. And I think Jenny had a question on the chat. I'm just reading the question.
Sorry, I was caught up in reading. There's a few coming through now. Yeah.
It might have been just a comment, I think, rather than
more a comment, really, just to say how useful that research has been in actual clinical practice. So I had a patient that actually went through the initial studies and was diagnosed with fructose intolerance through that, and he came back to me for advice on that, but also to manage some of the other IBS symptoms, and he was telling me about the experience and how helpful it had been. And then I've been able to use the data that's come out of some of those studies to be able to use that in my sort of practical explanation to patients of what's going on in the gut and why they get the swelling, why they get the bloating, why they get the rush and the urgency. So yeah, it's been really helpful for it.
I can't claim any credit for the
no.
It's mainly led by my luminal colleagues, which Lucas a big part of. So I guess, yeah, but I can thank them on their behalf. Thank you.
Yeah, send them a mental thanks.
And a comment from Dr. Clark as well, who's one of our radiologists in Queens about liver scan. The scan that we did is different from liver multiscan, although both of them rely on T1 relaxation time, but there are kind of important differences in terms of how the T1 was derived and how much of the liver that we measured, and there are differences. So we can do liver multiscan, but the data that I showed was different to the liver multiscan. But yeah, I agree. I think it's implementing these into clinical practice that would be the next step. Thanks, Chris.
Brilliant. I know we're a bit over time, but we think we've got 2 more questions, so I'm going to try and squeeze those in before the breakout room. So one of our direct to me, and it's asking what T-strength scanner is used. So that actually doesn't mean a great deal to me. So if you want to explain what that is, and then
I didn't get that question, Kate. Sorry, what was that?
What T-strength scanner is used?
Okay, so the initial work was done in 1.5 T, so 1.5 Tesla. So that just implies the strength of the magnet, but majority of the work now is being carried out in 3 Tesla scanners. But we are one of the few universities in Nottingham which have the strongest magnet, which is 7 Tesla, and they do advanced work, but that's more for experiments rather than routine clinical use, basically. Luca might correct me if I'm saying the wrong things. But
you've been absolutely spot on.
Something about sitting up scanners, we do have sit-up scanners. There are some of our lung doctors are doing some studies looking at how your diaphragm moves. They are using sit-up scanners. So there are new sitting up scanners in Sir Peter Mansfield Imaging Center. But most of the gut and liver work is done on scanners that are lying down.
I think that is most of the questions now, which is fantastic. I mean, if there's anything amiss, I do apologise. I will go through the chat and pick anything up and make sure that's answered following the session. But to avoid digging into any more of our breakout room time, that is what we're going to move on to now. So now I just want to thank you again for your presentation. I know you've got to dash off now, but just to thank you again for joining us this evening.
Any other further question, I'm happy for it to be emailed to me, so I can respond later.
Lovely, that's perfect. Oh, thank you so much again and thank you to all of our presenters today. All three extremely fascinating presentations, and I have popped in the chat that the slides and the recording of this session will be made available to everyone who's attended. So in case you want to peruse that after the session, and then if any other questions come to you, we are more than happy to answer any of those for you. I hope you all enjoyed your breakout room discussions. And I really hope you've enjoyed the entire event. What we'll be doing now is taking your feedback, your recommendations, your views, and any further questions that you've made within those breakout rooms, and collating them. And we will inform you of how we will be using those to improve research within the gastrointestinal and liver theme. So I just want to thank everyone again for your contributions today. It's so incredibly valuable, and I do really appreciate that you've stuck with us for the entirety of the event on this very humid evening. So what I'll do now is pass you back over to Guru, who will just thank you himself and give a summary of next steps, and that will let you go and enjoy the rest of your evening. So over to you, Guru.
Thank you very much. Yeah, I absolutely got more than what I could deal with immediately. So it is a bonus. What we are going to do now is that we have got a meeting next week of the team where all these key leaders will be there. I will want to see each of the breakout sessions, what actions there were. From the research program, as I said, that when we are able to open, we always will have an open meetings, and we will also have a showcase. And I can't foresee that happening till at least April or so next year. And we will open that. From your side, Kate and Andy, most relevantly, I've kind of appreciated beyond just participation in research, how we can include the community. I have actually got 3 tasks already from the breakout I have had. So I will come to you. I mean, I have been asked to, I have got some research which is ready to be disseminated. And I would like to approach a couple of ideas I have got here straight away. Karen told me to go to the schools to tell us how we can link food to the poo and what we do with the poo, and that will be stimulating both the children and the family. And Pramod asked me to really try and seek meeting the spiritual leaders in the community, particularly because food habits are innately related, at least in our culture, to their community activities. Many of them have religious overlaps. So, and with the research programs, we will certainly come to you and Andy to organize PPI-like events for individual groups and projects and programs. So that is what our actions are.
That sounds fantastic, Guru, and very exciting as well. I think you can see a lot of people nodding there.
Thank you.
And in agreement, especially me and Andy. So thank you, Guru. And again, thank you, everyone. Someone's commented in the chat asking if the outcomes from the other breakout rooms will be shared And yes, they will. We're going to be collating those, and then we will share them with everyone who's attended today for your information. So again, just want to thank everyone for your attendance today. It is so greatly appreciated. And again, to all of the fantastic speakers for your fascinating presentations. So we will be in touch. If you have any questions, I'm just going to pop a slide up now for you on how to contact us and find out more. But thank you very much, and we will see you soon, hopefully. Thank you.
Thank you very much.
Bye.
Lovely to meet you all. Bye.
Thank you.
Thank you.